Hi friends, as we have discussed core botany DL syllabus in our previous session, let us get started with the high weightage chapter in botany core syllabus of DL, that is plant anatomy and embryology. Plant anatomy means the study of internal organization of plants, be it root, stem or leaf. The study of internal organization of plants is called as plant anatomy. Before getting into the chapter, let us have the basic idea on all different types of plant tissues. Plant tissues are primarily classified into meristematic tissues and permanent tissues only on the basis whether they can divide or not. Those cells whose activity is only cell division, those cells whose activity only cell division are placed under meristematic tissues. Meristematic tissues, again on the basis of origin, on the time of origin, they are classified into primary meristem and secondary or lateral meristems. Primary meristems based on their position, they are again classified into apical meristems and intercalary meristems. Those meristems which occur at the apical region of root are called as root apical meristems. Those that occur at the apical region of the shoot are called as shoot apical meristem. Intercalary meristem occur in between two nodes, that is at the internodal region. They occur at the internodal region. And the main purpose of the primary meristem, be it apical or intercalary, is to increase the length of the plant. Secondary or lateral meristem will occur in the later part of plant life, whose only purpose is to increase the girth of the plant. Vascular cambium is a cambium which conducts the stellar secondary growth. It conducts stellar secondary growth. If the cambium is present in between xylem and phloem, it is called as intravesicular cambium, and if the cambium is present in between two vascular bundles connecting intravesicular cambium is called as intervesicular cambium. And the cambium which occurs in the cortex region, which conducts non-stellar secondary growth, is cork cambium. Vascular cambium is of two types and cork cambium conducts non-stellar secondary growth and occurs in the cortex region. Coming to permanent tissues, they have specific functional role. All the permanent tissues have specific functional role and they are classified based on their cellular composition. Simple permanent tissue has all the cells which are structurally similar. All the cells are structurally similar. Complex tissue have structurally dissimilar cells. Complex tissue has structurally dissimilar cells. Simple permanent tissue can be of parenchyma, collenchyma and sclerenchyma. Most of plant body is composed of parenchymatous tissue, and complex tissue includes conducting tissues like xylem and phloem. Xylem includes four tissues which are tracheids, vessels, xylem parenchyma and xylem fibers, wherein vessels are absent in gymnosperms. Phloem includes sieve tubes, companion cells, phloem fibers and phloem parenchyma, of which sieve tube cells are enucleated. And sieve tube cells, companion cells are also absent in gymnosperms. Rather, in gymnosperms, sieve tubes are present as sieve cells, companion cells are present as albuminous cells. Yes friends, now let us go into the details of meristematic cells, the cytological characteristics of a meristematic cell. Cell surface is isodiametric, means all cells are similar in structure and there are no intercellular spaces. There is no space in between the cells. Intercellular spaces are absent. Cell wall is thin and made up of cellulose. Cell wall is thin and made up of cellulose. They possess large conspicuous nucleus and they possess dense cytoplasm. Vacuoles are absent, plastids are absent and endoplasmic reticulum is poorly developed. The cells are devoid of stored food also, and meristems have capacity of perpetuation and ability to divide. They have the capacity of renewing the cells, cell renewal, and they have the ability to divide. The main characteristics which we have to remember is the cells are isodiametric, no intercellular spaces, and cell wall is thin, made up of cellulose. They possess dense cytoplasm. Vacuole is absent, plastids are absent and endoplasmic reticulum is also poorly developed and there is no stored food material. These points are very important friends, we have to remember this. Okay. Next, let us observe the cytological characteristics of secondary meristems or lateral meristems. Here secondary meristems that we have discussed just now friends, what are they? Like vascular cambium, cork cambium, these are secondary meristems, wherein the cells are long and spindle shaped. The cells are long and spindle shaped. Cell wall is thick, not like the primary meristems where cell wall is thin and made of cellulose. And here in secondary meristems, ergastic substances are stored, like starch, tannins are present in the secondary meristems. Fine. So, these are the characteristics we have to remember with respect to the cells of secondary meristems. Next, the classification of meristems are very important. Now, we are going to learn that in detail, wherein we have 5 bases of classification. 1, based on position, we have apical meristem, lateral meristem and intercalary meristem. Based on the method of development, we have promeristem, embryonic or meristem. Next, characteristics of initial cells, primary meristem and secondary meristem are present. Based on the functions, they are classified into protoderm, protocambium and ground meristem. Based on the cell division and growth pattern, they are classified into mass meristems, rib meristem and plate meristem. Let us understand this in detailed way. Like where is the location, what is their function. Fine. So, based on the occurrence or position of the plant, like apical meristem, based on the occurrence or position, we know apical meristems are at the apices, either at the tip of the stem. It contributes to the growth of plant shoot, stem, leaf and flowers. Root apical meristems, it is present at the tip of the root. It facilitates the growth of root system. See, here is the apical meristem and these are the intercalary meristems which is present in between the nodal region, and these which are at the lateral region and present and they occur in the secondary phase of the life, those are lateral meristems. If you look into the intercalary meristems, they are found at the internodes. As we have discussed, they are found at the internodes and the tissues are separated from the actively dividing primary tissues and occur in between apical meristems and permanent tissues. They occur in between apical meristems and permanent tissues. Okay, in the internodal region they are present. Fine. They occur in between permanent tissues at the base of the nodes. They occur at the base of the nodes. They are short lived. Intercalary meristems are short lived and merge with the permanent tissue later. They will merge with the permanent tissue later. They play important role in increasing the length of the plant body. Okay, as we have discussed, all the primary tissues will play important role in the increasing the length of the plant body. Most important thing that you have to remember is they are also responsible for increasing the size of leaves, flowers, fruits and examples. If you see internodes of grasses, leaf sheets of monocots and equisetum, and at the apex of the peduncle of taraxacum and gynophore of arachis. All these regions intercalary meristem is present. They are short lived and once they are matured they get combined or they get assimilated in the permanent tissue. Fine. And they are mostly found at the base of nodes and all these examples we have to remember. Yes friends, next lateral meristems. Lateral meristems, they are specialized meristems and they contribute in the secondary growth of plants, resulting in the thickening or widening the plant organs. This increases the girth of the plant, right? And the examples include vascular cambium, cork cambium and Phloem. Fine. So yes, so this is based on the position. Based on position, as we have already discussed in our initial chart, there is no much discussion about that. Now let us look into the meristems which are classified based on the stage and methods of development. First type is promeristem. Promeristem is undifferentiated and actively dividing cells are found at the tips of growing shoots and roots. These are promeristem. See, it is present over here. Promeristem is present over here, which includes actively dividing cells. Promeristem in an early stage of meristematic region is an early stage of meristematic region which is found at the embryonic stage, mainly responsible for giving rise to primary meristem. Promeristem gives rise to primary meristem. Promeristem occurs in the embryonic region and promeristem is a post-germination meristematic region found at the tips of growing roots. It is the precursor to the primary meristems and helps in forming the plant tissues during the primary growth. These cells are the initial precursor cells. They are what? Initial precursor cells that give rise to various tissues and organs in the plant as it grows. So if you look into this basic structure, promeristem is over here, which is near to the root initials. Okay, they form root initials, and at the surface of this region you can see protoderm, which is also called as embryonic epidermis. Protoderm, which is called as embryonic epidermis, and at the center you can see procambium, which is future central cylinder. Procambium, which is at the center, this is central cylinder. After that you have ground meristem. Ground meristem is future cortex, and to the peripheral regions you have protoderm, which is embryonic epidermis, and at the basal region you can find promeristem, which are root initials. Clear ma? Now, the promeristem is considered the earliest stage of development of the meristem. Most important thing we have to remember, friends, that is location and function. Okay, location is at the tip, which is at the apical region of root and shoot. Structure of the promeristem is they are small, densely packed, undifferentiated cells. They are small, densely packed and undifferentiated cells. Functions include primary growth, that promeristem gives rise to new cells contributing in the elongation of stems and roots. Second function is differentiation, that they eventually differentiate into more specialized cells. They eventually differentiate into more specialized cells, forming tissues like epidermis, vascular bundles and ground tissues. Okay, promeristems, once they are differentiating, they form into epidermis, vascular tissue and ground tissue. Clear, friends? Now, yeah, next is protoderm, which develops into future epidermis, that we have discussed. Procambium to vascular tissues, and ground meristem forms ground tissues, which includes the simple permanent tissues like parenchyma, collenchyma and sclerenchyma. Developmental role of promeristem is it acts as the foundation for development of plants of primary body, including leaves, stems, flowers and fruits. The next type is embryonic meristem or meristem. The embryonic meristem is nothing but these are group of undifferentiated cells which are present in the early stages of plant development. What are this? These are group of undifferentiated cells which are present in the early stage of plant development, specifically within the embryo. They are present within the embryo prior to the seed germination. They are present in the embryonic region. That is why they are called as embryonic meristems or Meristems. Yes, if we look into the functions or the activity they perform is undifferentiated cells. These are undifferentiated cells. Like other meristems, embryonic meristems consists of cells which are capable of division and differentiation, and most important feature of embryonic meristem is totipotency. Embryonic meristems can give rise to many different type of tissues which are structurally and functionally different. They can give rise to any type of the cell, that is totipotency activity. It is done only by the embryonic meristem or meristem. And importance of embryonic meristem is initiation of growth. It will initiate the growth during the embryonic stage, and as it is located within the apical region of developing embryo, this play crucial role in initiating of growth. Next, establishment of body plan. As the embryonic meristematic cells are totipotent, they can give rise to any different type of cells. So yes, they can establish the body plan of the plant body. If you look into the shoot apical meristem, which is over here, this is in the embryonic region, shoot apical meristem, root apical meristem, wherein this is the embryonic axis and these are cotyledons. Okay, the outer region develops to protoderm, middle ground meristem which gives rise to cortex, and this is the procambium. Clear? So what are the main function that you have to remember with embryonic meristem is those cells are totipotent and they establish the body plan, they initiate growth in the plant body. Now if we look into root apical meristem and shoot apical meristem are also part of embryonic meristem. Both root apical meristem and shoot apical meristem, just we have seen, right, those two are a part of embryonic meristem only, and because of the totipotency it involved in the entire plant growth and development. Okay, post embryonic phase you can see the primary plant body. Okay, during the time of seed germination we can see the primary plant body which includes root apical meristematic region and shoot apical meristematic region. From the central region periphery we have three different layers of shoot apical region. Okay, wherein central zone is at the periphery, rib zone is at the center, and towards the sides we have peripheral zones. Towards the sides what do we have, friends? We have peripheral zones. And root apical meristem has quiescent center, above which we have stellar region, and towards the sides we have endodermis, cortical region, and towards the surface epidermis. This is the basic structure of root apical meristem. In the coming slides we will understand it more clearly. So if we look into the basic differentiation, what all we have discussed, that and all are present here, friends. You can take a screenshot of it if possible. Just now whatever we have discussed, it is given in the comparative tabulated form. Okay. Now based on the nature of initiating cells, they are further classified into, just a second, based on the nature of initiating cells, they are further classified into primary and secondary meristems. They are further classified into primary and secondary meristems. Yes. So as we have discussed in the shoot apical meristem, just a second. Yeah, in the shoot apical meristem, here we have central zone, and bottom towards the center we have, towards the peripheral region we have central zone, and at the center below we have rib zone, towards the sides we have peripheral zones. Keep this in your mind. And here is the region of leaf primordia which gives rise to leaf structures. Okay. Coming to shoot apical meristem, wherein central zone which is at this region, okay, central zone will give rise to stem cells, okay, whose activity is continuous division, okay, and peripheral zone which is at the sides, they perform organ initiation. Peripheral zone conducts organ initiation function. Rib zone includes stem formation. These three functions are at most important, friends, you have to remember. Okay. Central zone will give rise to stem cells whose activity is only division. Peripheral zone, which are at the sides, they conduct organ initiation, and rib zone, which is at the bottom, they conduct the stem formation. They are involved in stem formation. Yes. Now, if we look into here, just a second. Yes, central zone will give rise to stem cells, correct? How they are giving rise to stem cells? Here it has a signaling pathway. It has a signaling pathway that is called as clavata-Wistel signaling pathway. It is what pathway, ma? Clavata-Wistel signaling pathway. When we are discussing about all the signaling pathways, we will discuss it in more detailed way. As of now, let us understand that from the central region, just a second, from the central region, we have the genes called Wistel genes. What genes, ma? Wistel genes, These genes from the rib zone, they will travel to the central zone. Okay, which genes will from the rib zone, they travel to central zone, whose only activity is to promote the stem cells formation or cell proliferation. Okay, which genes only function is stem cell growth and cell proliferation, stem cell proliferation. It conducts only that activity, and they strictly prohibit differentiation of the cells. They strictly prohibit differentiation of the cells in the central zone. And there are one more genes called as clavata genes. These genes will inhibit the Wish genes in cell proliferation. They will inhibit the Wistel genes in cell proliferation, and they promote differentiation. Okay, which genes will promote cell proliferation, stem cell proliferation, whose activity is inhibited by clavata genes. This will promote differentiation by inhibiting the activity of Wish genes. As of now, just remember this. In coming up slides where we are discussing about signaling pathways, we will discuss about it in more clear way. Second zone is peripheral zone, which is at the sides of the central zone, which is involved in the organ initiation. Cells derived from the central zone, they will migrate to the peripheral zone, where they differentiate to form leaves, lateral shoots and floral organs. What all this zone is forming is very important, we have to remember. Actively dividing cells contribute to organ primordia formation. Okay, those cells which are actively dividing in the peripheral zone will form organ primordia, and rib zone is mainly involved in stem formation, which lies beneath the central zone. Okay, rib zone lies beneath the central zone and its activity is mainly stem formation. It generates cells that contribute to elongation and thickening of stem. Okay, it conducts mainly in the elongation and thickening of the stem. Key characteristics that we have to remember with respect to initiating cells are, they are small and undifferentiating, and they have high mitotic activity. When cell division is very fast, respiration rate also is very high. Okay, high rate of respiration. High rate of respiration is seen. Okay, and maintains the ability of self-renew. Undergo asymmetric divisions to give rise to differentiated daughter cells, and responsive to hormonal and genetic regulation, which includes auxin, cytokinin and NOX genes. About NOX genes we are clearly going to discuss in the signaling pathway session. Okay, so don't miss that. Now, cork cambium or phellogen. We all know that this is the secondary meristem which occurs in the cortex region. So, in the cortex region, the cork cambium, which is also called as phellogen. Okay, cork cambium, also called as phellogen, will divide and form new cells. Those cells which are formed, those new cells which are formed towards the epidermis are called as cork cells. Cork cells or phloem. They are called as cork cells or phloem. Those cells which are formed towards the endodermis, those new cells which are formed towards the endodermis, they are called as secondary cortex cells. They are called as secondary cortex cells or phelloderm Okay, phloem, phellogen and phelloderm, together we call it as periderm. What do we call it as, friends? We call it as periderm. This is the basic information that all of us know. So, here initiating cells, which are nothing but phellogen initials, cork cambium initiating cells, they divide and form phloem or cork cells towards the outer side and phelloderm towards the inner side. And key characteristics of these initiating cells, initiating cork cambium cells, are self-renewing and actively dividing to ensure the continuous secondary growth. Secondly, they respond to hormonal regulation and they maintain asymmetric division to produce vascular tissues or protective layers, and they are present in woody plants and some of the herbaceous perennials also. Okay? Yes. So, here if you can see in this diagram, this is vascular cambial ring which is present in dicot stem, which promotes stellar secondary growth. This is cork cambium, wherein it forms a peridermal region which includes three tissues: phloem, phelloderm and phellogen. Okay? Next, fourth type, which is based on the functions, where the meristematic tissues are divided based on the functions. It is the scientist Haberlandt who recognized three categories of meristems on the basis of their function. First is protoderm. We know protoderm lately differentiates to what, friends? Epidermis, right? And procambium will lately differentiate into vascular bundles or conducting tissues, which is also called as central cylinder. Next, ground or fundamental meristems include all the cortex region, ground region, all the cortex region, which includes hypodermis, okay, which includes hypodermis, endodermis, cortex and all. Okay. So, yes, this is the clear diagram where you can see protodermal region, ground tissues, procambium, shoot apical meristem, and here if you can see leaf primordia, this is younger leaf primordia. Here it is older leaf primordia and this is younger leaf primordia. These are root apical initial cells. Yes. Here if you look into this diagram, wherein protoderm will differentiate to epidermis, ground meristem will differentiate to pith and cortex, wherein cork cambium further forms cork and phloem. Yes, procambium will form or differentiate to conducting tissues like primary xylem and primary phloem, which further is differentiated by vascular cambium into secondary xylem and secondary phloem. In root if you see protoderm, ground meristem and procambium. Protoderm to epidermis, however, ground meristem into cortex, procambium into vascular cylinder. Of course, here cork cambium, vascular cambium, again they lead to the formation of cork, phelloderm, secondary xylem and secondary phloem. All the points we have discussed already, but here it is in a tabulated form. If you want to take a screenshot of it, you can. Yes. First, protoderm, it gives rise to epidermis and epidermal tissue. Location is in the outermost layer of shoot and root apical meristem. Functions include, they develop into epidermis, which is the outer protective layer of the plants. Next, it is differentiation. How this is differentiating? They form certain specialized epidermal cells like guard cells in the leaves, trichomes in stem, root hairs in roots. Guard cells in leaves, trichomes in stem, root hairs in root. Clear? And the importance of protoderm is it protects inner tissues from the water loss, pathogen and mechanical injury, and this regulates gaseous exchange and transpiration as well. Procambium. Procambium, this gives rise to central cylinder or the conducting tissues. It is composed of narrow and elongated cells. This occurs in the central region of the plant body at the center. Okay, it is located in the vascular regions of the apical meristem, whose function is it develops the primary vascular system, that is primary xylem and primary phloem, wherein primary xylem it forms two different types of tissues, that is protoxylem and metaxylem. Protoxylem and metaxylem, whose occurrence in root. In root it is exarch. In root it is exarch, where protoxylem is towards periphery, metaxylem is towards the center at the pith. Whereas if you take in stem, it is endarch. It is endarch, where metaxylem is towards the periphery. Where metaxylem is towards the periphery, it is endarch, where metaxylem is towards the periphery and protoxylem is towards the center. This is the arrangement. Same pattern of arrangement you will see with respect to phloem also, which has protophlloem and metaphloem. In protophlloem, sieve tubes are narrow. In metaphloem, sieve tubes are broad. Okay. Basic differentiation, that is primary xylem will conduct water and minerals from roots to the shoot. Primary phloem will transport sugars and nutrients from leaves to other parts. Vascular cambium, in some plants a secondary meristem that contributes to secondary growth. In what plants does secondary meristem, that is vascular cambium, is formed? Gymnosperms and dicotyledons of angiosperms. The main importance is this ensures the mechanical support and transport of water, nutrients and food. Okay. Next, ground meristem. Ground meristem will give rise to ground tissues and medulla, whose location is there present between protoderm and procambium. Okay. The cells of this meristem are again isodiametric and thin walled. Their function is they forms the ground tissue system. Ground tissue system includes cortex, pith, medullary rays. To be more clear, ground tissue system includes all the type of cells except epidermis and vascular tissues. Except epidermis and vascular tissues, it includes all the types of tissues which are present in the plant. Differentiation: parenchyma, collenchyma and sclerenchyma. Parenchyma, of course, we have discussed, right? Most of the plant body is composed of which type of permanent tissue? That is parenchyma. They are thin walled. The cell wall of parenchyma is made up of cellulose and they conduct the functions like storage. If they have chloroplast, they perform photosynthesis. Then they are called as chlorenchyma. And secretion. In aquatic plants, the parenchymatous cells have air sacs in them which perform the function of buoyancy. And then if they are performing the function of buoyancy and if they are in aquatic plants, it is called as aerenchyma. Next, collenchyma. It provides flexibility to the tissues like petioles and mechanical support in the young plants. Their cell wall composition is cellulose, hemicellulose and pectin. Then if you look into sclerenchyma, they are thick walled and they have dead cells, okay. They are thick walled which is lignified. Their cell wall is thick and filled with lignin, they are lignified and they strengthen mature plant parts. Coming to the importance, they act as storage site for starch, proteins and water, and they provide mechanical support and photosynthetic in function in some cases, okay. In some cases they are also photosynthetic but rare, it is very rare, okay. Most of the photosynthetic function is performed by parenchymatous cells only. You will call it as chlorenchyma if it has chloroplast in it. Now, everything what we have discussed is in a tabulated form. You can take a screenshot of it if you want. Now, if we look into the final type or the basis of division, that is based on growth pattern of meristems. It is based on growth pattern of meristems. Meristems are classified into three types based on their plane and pattern of cell division. First is mass meristem, second is a rib or file meristem, third is plate meristem. Mass meristem will increase the volume of the cell and it divides in all the planes. Mass meristem will divide in all the planes and it will lead to the increase in the volume of the plant, okay. Cells divide in multiple planes, that is in all directions. There is no fixed orientation of division in mass meristem and they form thick and bulky structures in the plant. They form thick and bulky structures in the plants. And then examples of mass meristem, occurrence of mass meristem include development of embryo during early development. From embryonic meristem if you take, they are totipotent and divides in all planes through mass meristem. This is one example. And formation of pith and cortex in the stem, pith which is also called as medulla, okay. This is present in ground tissue system. Formation of these two tissues are also example of mass meristem which divides in all the planes. Growth of storage organs like tubers in potato which is a stem modification, underground stem modification, and bulbs, those are onions, okay. This growth of storage organs is also an example for mass meristem. The main function of this mass meristems include expansion of plant tissues and increase in the mass. Then importance for storage, mechanical support and overall plant volume expansion is done by mass meristem. Then comes rib meristem which increases the length. This is also called as columnar growth, okay. They mainly increase the length of the plant, those are rib meristems. Cells divide only in one plane, producing long rows and columns of cells. They are found in the elongating regions, of stems and roots. They are also called as file meristems because cells are arranged in vertical files like this. As cells are arranged in vertical files, they are also called as file meristems. Like this the cells are arranged in the vertical planes, that is why they are also called as file meristems. Examples of rib meristems or file meristems are, see, the columnar growth is formed because of rib meristems, fine. Yes, formation of vascular tissues, xylem and phloem in the roots and stem. Growth of palisade parenchyma in leaves. Root elongation through root apical meristem. All these are examples for rib meristems which are columnar meristems or file meristems, okay. Functions include, they help in elongation and axial growth and they contribute to the vertical expansion or extension of the plant organs. They contribute to the vertical extension of plant organs. Yes, the third type are plate meristems. This will increase the surface area of the plant organs. They will increase the surface area of plant organs. They divide in two planes. How many planes? Two planes. They increase the surface area and thickness. They produce flat and thin meristems. They produce flat and thin meristems like this, and cells remain arranged in a sheet like manner. Okay, cells are arranged in sheet like manner, those cells which are produced due to the plate meristem, that is two plane division. Okay, examples include growth of leaves in width and formation of epidermis is due to the plate meristems. Expansion of petals and other thin plate organs is also due to plate meristems. Yeah, functions of plate meristems include they help in lateral expansion, that is widening. Okay, as how rib meristems are giving columnar extensions, they help in widening. Just a second. They help in widening, that is lateral expansion, and they are essential for formation of thin broad structures like leaves to maximize photosynthesis. These meristems are very much important. Plate meristems are very much important because they increase the leaf size, they maximize the leaf size, and thereby maximum photosynthesis is conducted. This is the tabulated form of all the three types of meristems. You can take a screenshot of it. With this, I am going to end the first session of the chapter Plant Anatomy and Embryology. In the next session, let us have a brief discussion on the theories and then signaling pathways. Thank you.